Welcome to this edition of Archaeological Sciences interview sponsored by Society for Archaeological Sciences, also known as SAS. I am Ahana Ghosh, a doctoral student in archaeology at Archaeological Sciences Centers and the Humanities Social Sciences, IIT Gandhinagar, and a student ambassador for Archaeological Society for Archaeological Sciences. So today's our guest of honor is Professor Eleanora Reber, from Ceramic Residue Analysis Laboratory, Department of Anthropology, University of North Carolina, Wilmington. Before proceeding further to the interview, I want to convey my sincere thanks and regards to Professor Reber for her time and support. So we can now begin the interaction, Professor Reber.
Looking forward to it.
So in the beginning, I keep it more informal. In the beginning, I really want to ask you, as you're a ceramic residue analysis specialist, so when you started your career, the field of ceramic residue analysis was just growing. So what really led you towards studying the residues, and how your academic background shaped your above endeavor?
Well, I mean, I did it sort of differently in that I was originally an archaeology student. I chose my undergraduate school based on archaeology, and I went to a little school called Beloit College in Wisconsin. And my freshman year, my first year at Beloit, I took a chemistry class just because my chemistry in high school hadn't been good. And I guess I did pretty well because the professor, Dr. Brock Spencer, actually asked if I'd be part of a 400-level seminar on learning how to use gas chromatography mass spectrometry. And of course, I was very flattered and said yes. And I had a really good time. I learned a lot. And I realized that if I wanted to combine chemistry with archaeology, there were ways to do this. I read about it as part of my seminar. And so I tried a few things, and I realized that the GCMS, the gas chromatograph mass spectrometer, could be really useful in residues, and I already knew how to use it. And I was already very interested in issues of diet. So I just combined the two. And so I wound up double majoring. So I have a degree in both anthropology with the focus in archaeology and a bachelor's of science in chemistry. And I just sort of did both nearly all the way through, which I guess has been useful. And in my career, I've been pretty two-track. So I do residues here at the Ceramic Residue Lab where I am now. But I also do excavations, mostly around the United States, starting in Illinois. But recently I've been excavating in North Carolina. So I still kind of do both field excavation and archaeological chemistry.
So proceeding to my next curiosity is about what kind of support have you received from your mentors during your PhD days at Howard University? You have done your PhD from Howard. So what kind of support and motivations you have received from your mentors?
Well, I've been pretty lucky in my mentors through the years. My PhD supervisor was Professor Nicholas van der Merwe, who's originally from South Africa. And he was he was on this archaeological chemistry side. And so he would regularly kind of support me and help with lab work. But I also had archaeological mentors and, residue mentors as well. So that in terms of excavation, I was really helped out by Dr. John Kelly of the Washington University at St. Louis, who really helped me do my first excavation around the St. Louis area and helped me learn how to excavate in that part of the country. He wasn't on my dissertation committee, but he was really, really helpful moving forward. Also, I learned how to do residue analysis from both Carl Heron and Richard Evershed, who sort of served as my residue mentors. So although neither of them was actually on my committee either, they were both also, really essential. And then on my committee, I learned a lot from, from people who work specifically in chemistry, who taught me a lot about geochemistry in particular.
So as you spoke about South Africa, I really want to hear about your experience in South Africa, where you're learning this mass spectroscopy. As you told me when I met you about your different stages of learning mass spectroscopy at South Africa. I really want you to throw light on it.
Okay. Well, I mean, when I was in South Africa, I was really learning how to use not compound-specific mass spectrometry, but sort of your normal stable isotope mass spectrometry, normally for carbon, but the lab there also did, of course, nitrogen and oxygen as a standard. And Professor van der Merwe, who was my dissertation supervisor, had a joint appointment with the University of Cape Town. And he really wanted me to do some of my preliminary mass spectrometry work in his lab at South Africa. So when I wrote my NSF dissertation improvement grant, which included my time in Bristol, we also had an addendum where I flew into Cape Town and did 3 or 4 weeks in the mass spectrometry lab there, pretty much prepping bulk samples. So this is like a long time ago now. And people were just kind of learning, like compound-specific mass spectrometry was very new. So as a backup, because we weren't sure how well this compound-specific stuff would work, it worked really well. We decided we'd better do some bulk work as well. So when you're doing bulk mass spectrometry on residues, it's quite picky. So what you do is you extract the residues from the ceramic as usual, giving you, the tiny little bit of lipid in the vial once it's blown down. But then to get the carbon isotope and possibly the nitrogen isotope mass spectrometry data out of it, you put that little bit of lipids into a small tinfoil cup, and then you blow it down like a Carlo Urba cup, for those who've done it. You very, very carefully blow it down because they're very light tinfoil cups, and you're blowing it down in a nitrogen blowdown apparatus. So if you get the nitrogen stream too heavy, it tends to just blow the tinfoil cup away, and it goes sailing across the fume hood, and it gets contaminated, and you have to start all over again. And the tinfoil cups are, at least the ones I was using for the quantities of residue I had, were very small because it was a small amount of residue. So they were perhaps, 3/4 of a centimeter in height, and maybe 1/4 of a centimeter wide or thereabouts, very small. So you had to be very careful with the nitrogen. But if you did successfully blow the solvent out of the residue, you then had kind of a lipid coating on the inside of the tinfoil. And then you could simply put it in the Carlo Urba auto sample for the mass spectrometer. And then you could get a bulk carbon and nitrogen isotope reading for your entire residue. But of course, it wasn't compound specific, so it was prone to be contaminated by plasticizers, of course. The nitrogen was from proteins, which we weren't picking up in the residues. So it's a little problematic, but it was an extra data stream. And again, it was pretty early days, so we weren't really sure how the compound specific work how effective it would be. So we wanted to back up pretty much.
So now going deeper into your research, so what you have done and achieved so far, what are the pertinent insights or aspects you aim to reflect on? And I also want to know, what kind of functional group do you prefer to look into? And what are the most problematic biomarkers are to deal with? So I have divided this interaction in parts. So
yeah,
I'm really curious to know about this.
Okay, so for people who have a lot of background already on ceramic residue analysis, that the interpretation is pretty time consuming, and I'm not going to go back into the serious background. But one of the things we all struggle with is this concept of sample size, right? Because very often, as an archaeologist, I want to know everything, right? So if I've got a site and I've got, 5 different types of pottery, I want to know what's in all of them. But if I send only one sample of each type of pottery to my lab, my sample size for each type of pottery is 1, which isn't very good, right? So, that brings us to the concept of the functional group. What's the best sample size minimizing variables to tell us what's in a particular pottery type? And I think the functional group is really more of a conceptual idea. So it's based on whatever the archaeologist is interested in. It usually sort of reflects with a pottery type, but it could presumably, it can also answer questions such as, did residues survive in these ceramics? In which case the functional type includes everything you sample. But usually it's like a particular pottery form or type, or pottery found in certain contexts. And I think really 10 is pretty close to the minimum for a functional group. I mean, I've gone down to 7 and it's hopefully okay. But I personally feel better at 10. And that just kind of gives you a better grouping. So if, only one of those 10 samples is showing any residues at all, I'm wondering whether maybe that ceramic type wasn't used much to process lipid based, foodstuffs or resources. But then if all of them, all 10, contain, fish, then I begin to think, well, this pottery type was mostly used for fish or for dairy or something like that. But it does mean that if you've got a variety of different compound or a lot of different pottery types, you wind up with a much larger sample size to send to residue analysis than maybe people wanted. A standard, what most people send to a residue lab for, a pilot project or, what I sometimes call a fishing expedition is really about 10 to 15. And it's just kind of traditional. I've talked to my friends in paleoethnobotany and they tell me about the same, like 10 to 15 is what people send them. But given the functional group problem, unless they send me only one type of pottery from only one context and time period, all I can usually tell them is that residues were preserved or were not preserved from their residue or, from their archaeological site. If they really want to know what was in pottery type A from context B, we need about 10 pottery sherds from that specific, from pottery type A and context B. And if we have pottery type A and context A, and they're interested in differences between contexts, I need at least 20, probably. So yeah, you kind of have to think as an archaeologist exactly what question you're interested in answering. And then you have to decide, how important that is and how much you're willing to sacrifice in terms of, destroyed pottery sherds, because it is a destructive analysis. And also in terms of, how many sherds you want to send, what information isn't going to be measured. So if you send me 10 sherds from pottery type A and context A and 10 from pottery type A and context B, you're learning nothing about pottery type B. You sort of pushed it overboard, right? So you have to decide what's most important to you and whether you can give yourself another 20 sherds or not, because sometimes you can't.
So I want to just know that what are the most problematic biomarkers?
Right.
That is a big problem even I face when I try to interpret with my samples, that there are different type of contaminations.
Yeah, so, as you know, biomarkers are these compounds that specifically tell us what resources or types of resources are present in a residue. And the good news is some things are biomarkers for contamination, which means when there's contamination present, but of course it means you know how much contamination there is. I would say the most common biomarker for contamination, hands down, are the phthalates. So these are a whole series of plasticizers that are found in plastic bags. They get into everything. I think they're just chemically, they must be prone to bonding with ceramic, because all the normal cleaning procedures we do to remove contamination basically don't work well on phthalates. They pretty much will always
even
show
running
up.
a plank also. Even running a method plank also.
Mm-hmm. Well, the good thing about phthalates is they come explicitly from modern plasticizers, right? And they don't match very much in the natural world. So if you do find phthalates, which you're very likely to do if you ever stored your ceramics in plastic bags, which is why I discourage this, but even so, they kind of get around. You'll know there were some plasticizers that your pottery came in contact with, but it may not actually destroy the meaning of the interpretation too much, because they don't come from anywhere else, right? They're not going to change, they're not going to give us a false positive or a false negative or anything like that. The things that cause the most damage to interpretation are honestly probably cream sunscreens.
Yeah, that we faced.
Yeah, the problem being that,
recently.
yeah, because most sunscreens, of course, you rub on in a cream. Yes, they're sprays, but most of us rub them on. And they're designed to absorb within human skin, right? So they also absorb into pottery. And when people touch things, they'll get into the residues. And the other reason that's a problem is sunscreens don't just contain your active sun blocker. They also contain all those things that help it absorb within your skin. And that it doesn't even get into like scents, botanicals, I don't know, things like that.
There should
So
be a strong sampling protocol, I think.
So yeah, I mean, and I personally like a cream sunscreen. I can't dig without one. But you have to be very, very careful if you know you're going to be sending your shirt to residue analysis, not to sort of rub your hands all over the shirt, particularly if you've already recently put sunscreen on yourself. Because many of the compounds in the sunscreen that are meant to absorb within the body will mimic some compounds of archaeological importance. And that means that when we find a biomarker for sunscreen, we have to start looking at the fatty acid composition of the residue. And we have to think to ourselves, I think that fatty acid, some of those fatty acids came from sunscreen, but we don't know how much. So we don't know. At that point, the whole fatty acid interpretation is probably unreliable.
Okay. Now, going into one of the most interesting papers, interesting work done by you, this paper, the conjecture of ancient vegetarians, is a very popular paper of yours among the greater academia. Can you discuss some experience of working with the samples you mentioned in that paper?
Right. So ancient vegetarians actually grew out of my dissertation research. So in my dissertation, I was sort of originally looking to my plan, which of course didn't survive completely my entire dissertation, was to see if there was a relationship between the presence of maize in pottery residues and the Mississippian pottery type, which is generally in North America tempered with shell and is thinner than the pre-Mississippian or non-Mississippian pottery. So we had some idea that cooking maize might require the shell temper and the thinner ceramics. And so we thought we might find a correlation between those two. But of course, in order to do that, I had to figure out how to find corn and ceramics. And I also needed to find, which was sort of what my dissertation mostly was about eventually. And then we also had to find a whole range of ceramics from around the American Midwest that included both sherds likely to have included maize and those that probably didn't. So to do this, I asked around. I was digging in the American Bottom region, which is around St. Louis in Illinois and Missouri at that time with, Dr. Kelly, who I mentioned. And so Dr. Kelly and some other really helpful colleagues in the American Bottom region, Dr. Pocketat largely, and Dr. Kelly, gave me a bunch of samples from the definitely Mississippian contexts there. So that was nice. I also collected some ceramics from the Illinois River Valley, thanks to the people at the Center for American Archaeology at Campsville. And then T.R. Kidder gave me access to some collections from the lower Mississippi Valley, where maize was kind of adopted later. And so we thought, well, maybe we'd see less maize down there. And I also had a few kind of strange sherds from Wisconsin that were there because my undergraduate advisor wanted to see what was in them. So it didn't actually have a major bearing, to be honest, on my dissertation. But we were interested to see what was in the ceramics. And the rock shelter actually had Mississippian petroglyphs on the wall. So we thought, well, maybe there would be corn. So I worked with all of these, and there actually wasn't huge amounts of maize present in a lot of these, which, if I'd known as much about functional groups then as I knew now, I would have realized this. Even though the rock art on the Wisconsin samples was Mississippian, my ceramics that I sampled was, I think, looking back on it a little earlier. So I couldn't have expected probably maize in Wisconsin that early. The lower Mississippi Valley sherds, we actually did find some maize there, but not much, which fits our archaeological knowledge of the lower Mississippi Valley and maize adoption. And the Center for American Archaeology sherds, most of them had actually been stored for some years, so lipid yield wasn't great for a lot of them. So that actually landed us pretty much on the American Bottom sherds, which was our largest collection, which had the most likelihood of containing maize. And so that was where we found the most maize. And as we looked at all these sherds, both the American Bottom ones and the lower Mississippi Valley ones, we realized, or at least I realized, that compared to my, my graduate student colleagues at Bristol who were working more with European pottery, they were finding a lot of animal-based type residues. So they were finding a lot of cholesterol, and the fatty acids, although fatty acids are notoriously hard to interpret, really looked more animal-based than anything else. Whereas I was finding, in general, a little more residue on the whole, but a lot less cholesterol, a lot more plant sterols and plant biomarkers. And the fatty acids in general, keeping in mind the problems with fatty acids, look kind of more plant-like, or possibly fish. And so it seemed to me that my overall collection, not just the things including maize, but the things that didn't include maize from the lower Mississippi Valley and even from Wisconsin, appeared to be containing an awful lot of plants. Hence the title of the paper, Ancient Vegetarians. And as I've done more work in North America, I've realized that there are a lot of things probably feeding into this, perhaps more plant-based residues in North American residues. I think part of it is that in Europe and Britain, obviously, there are domesticated animals and there's dairying. Whereas in most prehistoric North America, the only domesticated animal is dog, which is not commonly a meat animal. So meat animals in most of North America are pretty much hunted. And so, they're game, non-domesticated animals that are less fatty to start with. So even when the indigenous peoples are cooking meat, the meat is less fatty than, say, a cow or a pig, or a sheep or a goat, or most of the domesticated animals. And then further in North America, there's notoriously very large stands of nuts that are also very high in fat compared to most other plant foods. So I think we may in part be seeing the importance of nuts in the diet of at least, the Midwestern and Appalachian indigenous groups where there's a lot of nut trees. And then as well, there's the fact that maize, which is the primary domesticate from Mississippian times onward in North America, is higher in fat than, for example, wheat or barley, or many of the plant domesticates from Eurasia. So I think it's a combination of all these things. And as a result, the residues that I've worked on in North America have very commonly been predominantly plant. I mean, some meat, but on the whole, I really expect to see more plant-dominated residues than animal or mammalian-dominated residues. And I expect to see a slightly higher yield, I think, than many of my colleagues have reported in most of Europe, except in Scandinavia, where they get much higher yields, certainly, than I'm finding in North America. And this is largely environmental, I think.
So as you mentioned about your working with the team in Bristol, I really want to know about your experience in working with Professor Richard Evershed, one of the pioneers and the founders in this field. He's working for long years. So how was your experience, if you can describe in a nutshell?
Oh, it was really remarkable. At the time, as I mentioned, it was early days, and my advisor, Dr. van der Merwe, had said that if I wanted to find corn in residues, which I did, then I was almost certainly going to have to do compound-specific isotope analysis. And the only person doing this was really Professor Evershed. So Nick, Professor van der Merwe, sort of contacted Professor Evershed and asked if, he would be interested in my working in his lab for, I guess it was a year about. And Professor Evershed very kindly agreed to do so. And actually, helped me find a place to live so that I lived with Steph Dudd, one of his, I think, graduate assistants or postdocs at the time. So I lodged with her in Bristol. And at that time, and, since I believe it's got even bigger, it's just a massive facility all dedicated to residue labs. I don't know that there's to residue analysis. I don't know that there's anything like it in the world. He had a cluster of undergraduates, he had a lot of graduate students, he had postdocs. He had at the time, I think, 4 plain GCs with the FID. I think 2 might have been more GCMSs, and then 2 working compound-specific isotope mass spectrometers, which at the time was, just insane amounts of compound-specific isotope mass spectrometry. And then they had, very ample bench space, even though it was a little bit crowded because there were so many people. And the whole facilities, for the clean glassware, for the good water, for the adequate solvents. So I didn't have to worry with all that sort of the logistical aspects of setting up a residue lab, which, the devil's kind of in the details there, and that was all taken care of. So there, particularly under Steph Dudd and Ian Bull and a lot of other people there, they sort of refined my residue extraction. And I learned how to do the compound-specific isotope work, which eventually wound up helping us to find corn residues. But it was just a remarkable kind of grouping of really talented researchers who are all pretty much devoted to different types of residue analysis. There wasn't anything like it in the world at the time, and there still may not be. I don't think there is.
I really dream of visiting there, or if I even get an opportunity to work there one day. Yeah. So going to my next question, how this field has changed over time? What are these new additions that has been done in the corpus of residue research in every corner of the world? What do you think is the pertinent additions and the changes that happened? You have been in this field for last 20 years. So how you have seen the change?
Well, there have been a lot of changes. I think big picture, the analysis is much more accepted than it once was. At least in this country, CRM firms, the professional archaeologists will send their pottery out for ceramic analysis if they can find someone to do the work, which is actually a little bit difficult because there aren't that many trained analysts and interpreters, and we're all pretty much at academic locations. But aside from that, I would say there have been massive changes in certainly the compound-specific side. And then the changes in extraction have perhaps led to the greatest shifts in interpretation. So that kind of the baseline method of residue extraction involved solvent extraction, usually with a mixture of chloroform methanol. And that gave you what was called the total lipid extract, which include all the acyl lipids, so triacylglycerols and diacylglycerols. And we then processed those total lipid extract into a fatty acid extract and then a neutral extract, which gave us things like sterols and alkanes and alkanols. So anything that wasn't extracted by the chloroform methanol basically stayed within the sherd, and we didn't see it because it was still in the sherd. And further, that type of extraction was, it's all a little time consuming, but it also led to 3 separate instrument runs, each of which is, depending on your instrument methodology, between half an hour and 40 minutes in time plus cool down. So on the instrument side, even aside from extraction, that methodology takes 3 hours of instrument time per sample. And then you had to separately interpret each sample, which is at least probably 2 hours plus. So that's time consuming, right? So with some of the new extraction methodologies that people have used, using both alkali methanol and acidic methanol, you can extract out a lot more things from the ceramic so that less is left in the pottery when you're done extracting, and we learn more. And also particularly the acidic methanol extraction method that Correa, Sensio, and Evershed published a few years ago. You don't get the total lipid extract, so you don't see the composition of the acyl lipids, which sometimes is a bit of a problem. But on the bright side, it does give you essentially a single extract that you can run one time and interpret one time. And it also takes out a lot more things from the pottery. So as a side effect, it looks like a lot of the biomarkers for fish don't actually extract well with chloroform methanol. And so the massive expansion in identifying fish and aquatic resources and residues, if you look at it, actually follows directly along the change in extraction technologies, not just the acidic methanol extraction, but also alkali methanol or alkali extraction does something similar. So pretty much everyone finding large amounts of fish is usually using one of these non-chlorophore so extraction techniques, which in addition tend to be faster. So if you use acidic methanol, that's one instrument run at about 1 hour, maybe a little bit less. Interpretation is more time consuming because the extract is much more complicated, but it's still faster than 3 extracts. So I'd say it's probably cut analysis time about in half for me, probably more for other people.
Okay. So now I really want to know that if you're comfortable, can you share with us about the interesting projects you are doing in your lab, a bit about it? We have discussed this during my visit. So I really want to know some about it, or experimental methods you are using now.
Okay, so I actually have a lot coming into the lab right now. We're busy. But we have a whole series at the moment of experimental sherds that have been used to process beans, Phaseolus vulgaris, along with other various resources. And we're looking to see, firstly, what lipids from the beans will show up in the ceramics, and then perhaps seeing if we can maybe find a way to, look for a biomarker. We'll see if that works. At the same time, we also have a whole lot of sherds from the Stallings Island sites. That's Emily Barks's work. She just got a NSF dissertation grant. I think I can probably safely say that without divulging any of Emily's secrets. We have a large number of sherds from Stallings Island that we'll be working on together, which is really exciting because Stallings Island is one of the very earliest pottery types in the southeastern United States. And one of the great questions in terms of ceramic development in the Southeast is, what were these pottery sherds used to process? Was it a single resource, like, for example, fish, as seems to be the case in some parts of the world? Or is it more of a mixed processing of foodstuffs? So the preliminary analysis, we did about a functional group of about 10 or 12
So it's
sherds.
very diversified. You're working on diverse kind of samples.
We are, yeah. And then we have some salt pans from Tennessee that are 2,000 years later, but that's a different project entirely. We wish to see if they're truly salt pans, they were only used to process salt and water, and there should be no residues in them. If they weren't salt pans, there should be residues. So for that study, the functional grouping is relatively simple, right? Just salt pans. If there are any residues, that will actually answer our research question. That's nice and relatively simple for our group.
Okay. So I have worked with you and the lab you're sitting behind. I really miss this place. You have been really patient in teaching me how to do the extraction, and we have worked for days and discussed about residues. I bombarded you with a lot of questions about residues. So you're really a wonderful collaborator and mentor to work with. So how was your experience in working with me on this South Asian shirts, and especially on this, rather than me on the South Asian shirts? It was a new genre for you. And how do you what do you want to share about it?
Well, that was really interesting. I really enjoyed working with you because, you're so interested in residue analysis, and also because I've been interested in the Indus Valley for a while. When I'm not doing residues or excavating, I also teach archaeology. And I do teach the class in Egypt, Mesopotamia, and Indus civilizations here at UNCW. So over the years, as I work on my classes on the Indus Valley, I have wondered what the ceramics were used for. And, what would happen if we had some residue analysis. And so when you showed up with, pottery sherds from the Indus Valley, I was very excited. And so I'm really interested in your projects. And it's always good when we have someone who wants to learn the extraction and the interpretation, who's also an archaeologist.
It's my pleasure to learn from you. I hope I keep on continuing learning from you.
Well, I think it's important for people to have both the excavation and a little excavation, as well as their archaeological science work, just so they know what the archaeologists are getting up to sometimes.
So coming to the last leg of our interview, my last question to you. What is your message for the scholars taking up residue analysis as a methodological pursuit in their research? So what is the message you want to send to them that who wants to take up residues?
Oh goodness, that's a hard one.
At the end, I came with the tougher
Yeah.
one.
Well, I mean, it's difficult because the usual method is it's a lot more complicated than you think, right? People tend to look at the papers and think to themselves, well, I can tell that this pot maybe contained deer. And sometimes, you know, with careful work on the biomarkers and compound specific isotope work, you can do that. You know, some of the work on dairy is just amazing, as is some of the work on fish, marine lipids, and so on. But if you're working on terrestrial normal residues, most of the time, the interpretation is quite tricky. In order to learn it properly, you probably can't just read a couple of papers and try it out with the chemist pal. You probably need to go work with someone who's done residue analysis and do something of a training or an apprenticeship, because the body of knowledge is pretty eccentric. It'll be different from what your chemist pal has done. They probably focus more on organic syntheses, or if you're lucky enough to have a natural products chemist pal, they'll be familiar with the chemistry, but only recently. And we're archaeologists, and all our chemicals are, you know, more than 100 years old, sometimes a lot more. And so it's more complicated than people think. And yet I don't want people to think it's hopeless, because it is a really exciting and useful area of study. It's just one that requires a lot of input. So in a lot of ways, it's very similar, I think, to paleoethnobotany or ceramic petrography. It's a method that requires a fair amount of training to get right, but nonetheless, it's really, really valuable and important to answering archaeological questions. So persist, I guess. But
So
yeah,
we
you won't
want
learn
more
everything
people
right
to
away.
come and join us in this field. And
Yes.
so we look forward to that. And we end this interview on that note. And thank you again, Professor Reber, for your time and this wonderful insights we got from the interview. I am taking a lot from this interview. Thank you very much. Thanks again. And we can end this interview here.
Okay, thank you very much.